Let's do a little bit of a recap here. Other than water molecules, the major molecules of living things are the macromolecules, really huge organic molecules. And there are four major types: proteins, carbohydrates, lipids, and nucleic acids. When we talked about the lipids, there's a hybrid of those ones, think of fats, oils, and waxes, and things like that. And mostly they're used for lipids. For energy storage, think of fat on your body mostly. That's its job, is to store calories for you. But it also functions as insulation and cell membrane. This next category, macromolecules, proteins, are pretty interesting because they do all sorts of functions inside living things, inside cells. Maybe mention first what their monomers are. All the macromolecules are polymers, meaning they're smaller organic molecules. Each of the four macromolecule types has its own particular monomer that it's built on. And for proteins, they are made out of a monomer called amino acids. And there are 20 different subcategories, 20 different types of amino acids that are going to represent like 20 different colored balls here. So proteins is a bunch of those amino acids that we all put together. Like this. I mentioned last week the average protein is around 400 amino acids long. Some are quite a bit smaller. The smallest one I've heard of that I ever read about is 8 amino acids long. And some of the larger ones are thousands and thousands of amino acids. 400 is more. And since there are only 20 types of amino acids, and the average protein is 400 amino acids long, you should infer from that that it's okay for amino acids to be repeated in a protein. Short protein here, you can see this amino acids repeated twice in a row and another time over there. All right, so what are the function of proteins? Well, that's a big question because they do a whole bunch of different things. Probably if you got a textbook, you can find at least a dozen descriptions of protein functions. But if I can sort of generalize a little bit, I'd say the proteins carry out all the tasks that keep cells alive and functioning. Proteins are not living things, they're just molecules, but in a way they act like living things. Proteins can move around, a protein can grab hold of some other molecule and change it in some way, or move it in and out of the cell. Whenever we're studying living things, and I say the cell does this job, or the cell carries out that function, the cell does this task, any sort of action or function going on inside the cell, you can think of as a protein carrying out that. Here's one. Thank you. You can think of a protein as carrying out those functions. And I listed some of your chemical reactions inside the cell, importing and exporting things out of the cell, that sort of thing. And in this cartoon here in the cell, I made the protein provide for the workers, because that's what they do. They actually carry out functions inside the cell. They're the workers in the cell. each protein is a specialist. Each protein only works with one particular molecule and always carries on the same function on that molecule over and over. This protein up here, his job, let's say, is to make that purple molecule. That's the only molecule we ever use. This protein right here, his job is to import this molecule right here. That's the only thing this protein does is import that particular molecule. Just absolutely specialist in each task. One task. The sequence of amino acids, what order you find amino acids in a particular protein. Think of that as the programming of the protein. What gives this protein its particular job is a sequence of amino acids. It's a different sequence of amino acids, so that will have a different function. You might wonder, well, how is it that the order of the amino acids within a protein gives the protein its particular job? We'll hold that question and we'll answer in just a few minutes. Okay, so proteins do a whole bunch of different things inside living things, but what I consider to be the four most important protein functions, maybe at least the four most important types of proteins are these receptors, transport proteins, enzymes, and structural proteins. And let's go down this list and talk about where you find these proteins, exactly what their function is. Well, actually, you tell me, this is a part of the cell. What part of the cell is this supposed to be? The membrane. The membrane of all cells is a phospholipid bilayer. That's what those circles with the two tail parts, the phospholipids. That's the hydrophilic part of the phospholipid, with the two tails hanging down to the hydrophobic fatty acid tail of the phospholipid. And all membranes are these phospholipid bilayers. So remember that the fatty acids, hydrophilic tails, I can't speak of the hydrophobic tails of the fatty acids are very hydrophobic. And so this interior part of the membrane is an extremely hydrophobic area. Right. Now, embedded within the membrane of cells is where you find these receptor proteins. And essentially the function of a receptor protein is to serve as the antenna for the cell, or maybe you might think of it as the sensors for the cell. These receptor proteins stick through the cell membrane, and part of them sticks outward from the cell on the outside of the cell like this. And their job is to detect for the cell what molecules are floating around. And so let's say this is the molecule that this receptor is supposed to detect right there. To detect the molecule, it has to bind to the receptor. The receptor proteins will have little eyeballs to spot molecules floating away, floating around far away. The molecule that the receptor is supposed to detect has to bind to the receptor for the receptor to detect it. But when the receptor does bind to the molecule, the part of the receptor that sticks inside the cell alerts the cell, warning, I'm detected by molecule, and then the cell gets the message that that molecule is present, and oftentimes the cell will have some particular response to that molecule pre-programmed. Now let's pop that molecule out of the receptor in one moment. Just a minute or two ago, I told you that each protein is a specialist. It only interacts with one particular molecule. And the same is true for receptors. Each receptor is a specialist in detecting just one particular type of molecule. And what makes each receptor a specialist is this area right here. It's called the binding site of the receptor. And the receptor is a specialist in detecting just this particular molecule because that's the only molecule that fits into its binding site. I think the next section of your handout, let me leave this receptors bit for just a second, I'll come back to it, but in the next section of your lecture outline, there's a little information here on the binding site. The binding site is a crevice or a fold or a gap, something that is in a protein where it binds the molecule it's supposed to be working. And the official name for the molecule that binds into the binding binding site is called the ligand of the protein. Kind of a funny word. I think the word ligand comes from a word to join together, because this pops in here and joins onto the receptor. And well, there it is right there. The crevice inside the protein when it binds the ligand is called its binding site. And here's the way to think of these binding sites. They are formed by the folds of the protein's amino acid chain. And just remember, every protein is a big long chain of amino acids. And proteins have a natural tendency to fold themselves up and sort of twist it back and forth in knots and turns and convolutions like that. And a good way to think about it is old rubber bands. If you've ever taken something out of a drawer that you haven't looked at in years, and whatever the thing is has been wrapped up by a rubber band, if you take a rubber band off and let go of the rubber band, it wads itself up. Or if you like fishing, if you have fishing line that's been wrapped around your spool of a fishing rod for years, and you take off that fishing line and let go, it wads itself up automatically so it can twist it really long. Well, what I'm saying is proteins do the same thing. They have a natural tendency to fold themselves up. And the binding site is formed by the folding of the protein. As long as the protein folds correctly, that binding site will form. And that's why, a little while ago I mentioned that the sequence of amino acids is what gives each protein its function. The sequence of amino acids determines exactly the shape of the protein when folded to. And so the sequence of amino acids determines the shape of the binding site, and then the shape of the binding site determines what the molecule of protein is going to be able to bind to. And so if the protein is folded correctly, the binding site therefore forms correctly. The binding site will be the exact shape of the molecule that the protein is supposed to bind to. The exact shape of the ligand. And you sometimes hear an analogy of locking